Good morning, everyone. I think for the last few classes, we were discussing about the introduction of viruses, and compared to bacteria, viruses are easy to study because in bacteria we have to learn the morphology, the pathogenesis, etc., the clinical manifestations. But some of the viruses we already know the clinical symptoms, and we already have suffered from it. For example, rhinovirus, chickenpox, etc. So viruses are really interesting to study when you compare between a virus and bacteria. So today's class is about the cultivation of viruses. So in the previous class, we were discussing about the structure, shape and the interactional characters. That is what is virion, what is the capsid, whether the viruses are enveloped or non-enveloped viruses. Then what is the virus? The viruses may be having the nucleic acid of two different types, that is RNA and DNA. Then what are capsomeres? And that is the initial introduction of viruses. What is actually a virus? What is virion? And let me have a recheck or a recap of the previous important points, that is capsid, capsomeres, nucleocapsid, virion, and how is the viruses destroyed. That is, viruses can be destroyed by various chemical substances such as sodium hypochlorite and also other autoclaving, etc. But when you are coming to the cultivation of viruses compared to bacteria, we can cultivate bacteria by artificial culture medium. That is what we have learned in our bacteriology classes. That is, we are cultivating bacteria in a artificial culture media, that is such as blood agar, McCongie, or nutrient agar. We have so many selective media to understand the different morphological characters, and all the different type of media that shows the bacterial cultural characters can be observed under the naked eyes. But when we are discussing about virus cultivation is quite difficult in a daily diagnostic basis. And when you are cultivating viruses, we have to be much careful and precaution to be ourselves affecting from the viruses and to have less contamination in our work. So in the cultivation of the viruses, we are having sophisticated methods recently or in our current scenario. But the earlier cultivation, what was previously used as an initial method in virology for the cultivation of viruses. So that is what we are discussing now or in today's our class. So by this class, as a learning outcome, you will be able to understand the different methods of cultivation of the viruses, what is the importance of viral cultivation, and how is the virus growth dictated by after cultivation. This is what we are going to learn in our today's class. So the initial cultivation method or the primary purpose of viral cultivation is what is the primary purpose? To isolate and identify the viruses in clinical specimens. And not only to isolate and identify the virus from clinical specimens, we can also prepare vaccines to prevent the various viral infections. And also, we are cultivating the viruses to understand their structure, what is their effect on the host cells, etc. And the most important thing to remember is that viruses are obligate intracellular parasites. So what is the important thing here to remember? They can be cultivated only in live cells. Okay. Also, we are having different functions for why we are doing. What is the purpose means? We need to isolate, we need to identify the structure of viruses, we need to prepare vaccines, we need to understand its morphology, we need to know what is the effect in other human cells when a viral infection occurs. But the most key point is that whatever happens, to understand the viral morphology, we need the live cells because viruses are obligate intracellular parasites. So they cannot be grown in an artificial culture medium. They need live cells. So what are the main cultivation of viruses, or what are the common methods used in virology to grow viruses? The first method is animal inoculation. The second method includes embryonated inoculation into embryonated egg. And the third method includes tissue culture. When we tell about animal inoculation, the common laboratory animals used for culturing viruses includes rabbits, guinea pigs, ferrets, etc. But the most commonly, although we have these three animals, the most common animal used in virology lab is albino mice. By inoculating into animals, we can study the viral pathogenesis or how does the infection occur, after how many days the clinical symptoms occur, when the viral infection is occurring. For example, chicken pox, after incubation period, what is the real first blister, where is the first blister appearing, what is the shape of the blister, what is the pathological changes in the skin. These can all be observed in animals by our naked eyes. That is the advantage of animal inoculation. But there we have to have so many ethical reasons. We cannot inoculate all the viruses into animals. So there are so many ethical complications also. So in order to study for research purposes, we are using albino mice in the virology lab to understand the morphology and pathogenesis. The different routes of inoculation into viruses. What are different routes of inoculation? The different routes of inoculation includes intracerebral, subcutaneous, intraperitoneal and intranasal. So there are so many routes of inoculation. The routes of inoculation are intracerebral, subcutaneous and intraperitoneal depending on the virus to be isolated. And once you inoculate the virus, the animal is kept under observation. Okay, we have to observe the animal for a few days to see the clinical symptoms. How does the pathogenesis occur? Where are the initial lesions of the infection occurring? And how is the progress of the infection? So after the what is the different route of, after the route of inoculation, we are keeping the mice under observation to study the pathogenesis, to observe the clinical symptoms, etc. And the second common method that is used to cultivate virus is embryonated egg. And here we can use either hen's egg or duck's egg. This method was first introduced by a scientist known as Goodpaster and Burnett in 1931. For this embryonated egg, we are using 7 to 12 days old embryonated hen's egg. We can either use duck's egg or hen's egg, but the age of the embryonated egg is usually 7 to 12 days. And here also we are having the different route of inoculations, because in a embryonated egg we need to do the candling first, because we have to identify where is the site the virus has to be inoculated. In a embryonated egg we have different sites of inoculation that includes chorioallantoic membrane, amniotic cavity, allantoic cavity or yolk sac. Okay, so in order to inoculate into these different routes, that is either you can inoculate your virus into chorioallantoic membrane or allantoic cavity or amniotic cavity. Before inoculation, what you have to do, you have to first disinfect the egg, and after disinfection we have to candle the egg. That is, candling is done to find out where is the site, and then a hole is made in the eggshell to inoculate the viral specimen, and this is done using a sterile syringe in purely aseptic conditions. Okay, that is the most important thing. Although you are having embryonated egg, before inoculation you have to disinfect the egg, and after that you are finding which is the site of inoculation, and you are making a hole in it, and once you make a hole in it, the viral suspension will be introduced into the shell using a sterile syringe. And after inoculation, the egg is kept sealed with gelatin, which is kept inside the incubator. And then it is observed for the changes. What are the changes that you see inside the egg? The different changes may be seen as pox or lesions. In case of a chicken pox virus, or when you inoculate into embryonated egg, there will be pathological lesions in the hen's egg, that is known as pox. And these pox can be identified by different serological test. So already I mentioned to you the different methods. Once again I am repeating. The different routes of inoculation in a embryonated egg includes chorioallantoic membrane, amniotic cavity, allantoic cavity and yolk sac. And different type of viruses can be inoculated. For example, chorioallantoic membrane. In the case of chorioallantoic membrane, we are cultivating herpes viruses and pox viruses. In the case of amniotic cavity inoculation, there will be different type of viruses, that is mumps virus and influenza virus can be inoculated in or cultivated in this type. That is amniotic cavity, we can cultivate mumps virus and influenza virus. And in the case of allantoic cavity, we are mainly inoculate to or culturing viruses to prepare vaccines to yield large amount of the virus. Okay, in the case of allantoic cavity, we get large amount of the virus. So normally we are cultivating it for preparing vaccines, because here we are injecting 8 to 15 ml of the viral suspension. And the common method of inoculation is yolk sac inoculation. And in this yolk sac inoculation, the different bacteria also can be cultivated, such as chlamydia and rickettsia. So in yolk sac inoculation, we can cultivate viruses as well as certain special type of bacteria. The common another method, so one is animal inoculation, the second one is embryonated egg, and the third method includes tissue culture. Tissue culture is mainly by three methods: organ culture, explant culture and cell culture. The three type of tissue culture used in virology lab is organ culture, explant culture and cell culture. What is organ culture? Here, specialized tissues will be used to cultivate virus. Specialized bits of organs, not tissues. Here, specialized bits of organs will be maintained in a tissue culture medium, and that is used for growing certain special viruses. Example of such virus grown in the organ culture is coronavirus. So the first method is organ culture. Small bits of organs are maintained in growth medium. Here it is used for isolating specialized viruses. Example of such viruses, coronavirus. The second one is explant culture. Here in explant culture, we are using fragments of minced tissue. This was a method which was previously used. It is not used nowadays. And the third method is the most common method used in virology laboratories nowadays, that is cell culture. In cell culture, we have three types, that is primary cell culture, diploid cell culture and continuous cell culture. When we tell about primary cell culture, these include the normal cells which are taken from organs of animals, and they are capable of limited growth. This type of culture is mainly used to isolate viruses or to cultivate vaccine. So what is the main function of primary cell culture? The main function of primary cell culture is used to isolate the virus or to cultivate viruses for vaccine production. Example of such cell cultures is human amnion or chick embryo cultures. In the case of secondary type or the diploid cell cultures, they can be subcultured for a certain limited number of times, and this is mainly used for the isolation of some fastidious viruses. Okay, so they can be cultivated for a few number of times, and they consist of diploid cells, and it is used for the isolation of fastidious pathogens. So the diploid cell culture is mainly used for the production of viral vaccines, especially rabies vaccine. So diploid cell culture is mainly employed for the viral vaccine production, example rabies vaccine. And last one is continuous cell lines. In the case of continuous cell lines, we are taking cell lines from the cancerous tissue because they will multiply continuously. So they are known as continuous cell lines. It is mainly also used for the growth of viruses, especially for vaccine preparation. So we have primary cell lines, diploid cell lines and continuous cell lines. The continuous cell lines are obtained from cancerous tissues. In the case of cancerous, when you take cancerous tissues, the virus can multiply in it indefinitely. That is why it is called continuous cell lines. Although we have all these three methods of cultivation, that is animal inoculation, embryonated egg and tissue culture, the viral growth detection is also important. So how can we detect the growth of viruses? The growth of viruses can be detected by different methods such as cytopathic effect, heme adsorption, interference, transformation, immunofluorescence, electron microscopy, and with the application of certain enzymes. Cytopathic effect means there will be some changes that may be occurring in the cell, that is known as cytopathic effect. Immunofluorescence need to be detected under fluorescent microscope. Electron microscopy also we need electron microscope help to see the changes. So heme adsorption means it involves the clumping of RBCs due to viral infection. The details of the detection of the viral growth will be discussed in the following class. I hope everything I have discussed today is clear to everyone. Are you able to understand? Is there any doubts? Now you can ask me any doubts regarding this animal or viral cultivation methods. Please raise if you have hands if you have any questions. Thank you and have a nice day.